Hello, everybody. Welcome to today's webinar in GNOME's new educational webinar series. This is Ben Salisbury speaking. I'm the Senior Vice President of Clinical Products here at GNOME, and I'm pleased to introduce today's speaker, Dr. Corey Nislow from the University of British Columbia, where he works in their Faculty of Pharmaceutical Sciences. He's also the director of the UBC Sequencing Center at Pharmaceutical Sciences. His talk today on leveraging in silico genomic panels to translate next-generation sequencing into clinically actionable information is going to be very interesting and informative. We are recording today's webinar so that afterwards you'll be able to get a copy of that, both the video as well as the slides. We have over 300 people registered for this webinar today, so we're excited to get going. On the next slide, Corey, wanted to let everybody know that if you have any questions during the webinar, please enter them in the GoToWebinar pane. So in the GoToWebinar pane, there are a variety of sections you can open up, and one is for questions. Drop those questions in there. For technical service, we'll get back to you immediately. And questions for Dr. Nislow, we will get to at the end of the webinar. We'll try to get to as many as we can, and those that we don't, we'll get back to you later in person. And with that, welcome, Dr. Nislow.
Wonderful. Thanks so much for the introduction, Ben, and I hope over the course of the next 45 minutes or so to convey to everyone the work we've started here at the University of British Columbia and our efforts to really translate genotypic information, combine it with phenotypic information collected primarily at community pharmacies, and use that information to improve healthcare and health outcomes for clients and patients. So as an overview for today's talk, I'm going to focus on the bottlenecks that we and most large-scale projects that are designed along these lines will invariably face. But it's not all doom and gloom. I'm going to highlight the ways we've surmounted most of these bottlenecks. So we'll talk about how one generates the data. This will be a review for most of you. Assembling the infrastructure to actually pull off a large-scale project that involves data collection, analysis, and delivery of results. And as a key part of that, the final bottleneck being managing and analyzing the data. And that's where the GNOME platform that we've adopted at UBC will come into play. Just a 30-second review of genetic variation for all of you in the audience who undoubtedly know better. The DNA replication machinery, thankfully, is extraordinarily accurate, but every once in a while, an error is made, and that error is the fuel of variation in evolution. And genetic variants can take a number of different forms. We're still discovering some of the types of variation in the forms of chromatripsis, etc. But for today, I'm going to focus on the single nucleotide polymorphisms and how we can use information about individuals' SNPs to hopefully guide drug therapy and avoid adverse drug reactions. So what can we hope to learn from studying SNPs? As I just alluded to, our primary goal, or one primary goal, is to identify SNPs that are associated with disease development. there are several large-scale projects in diabetes, heart disease, the association with variation and predisposition to addiction, as well as Alzheimer's and autism, and not to mention the numerous success stories with cancer genomics. One can also identify patients who will benefit from certain drugs. There are, in fact, drugs that cannot have any therapeutic benefit to certain genotypes, and so there's no utility giving patients those particular medications. And then finally, what our project is focusing on is being able to predict the differential response to drug therapies and using genotypic information to be able to adjust the dose of drugs to fit a particular patient's genotype. So as I said, I'm going to try to hit on 3 bottlenecks today. The first bottleneck historically has been to generate the data, and we all know the story of the Human Genome Project taking about 10 years, although the bulk of the sequencing was done in the last 2, and for 1 to a few individuals, the cost was $3 billion. And then the projections were that we should be near the $1,000 genome right about 2013 if the impetus from the National Human Genome Research Institute paid off. And in fact, it did. And right about now, we are at the $1,000 or so genome, depending upon how you do the math. And so I'm not going to look to the future and rather make the case that the time for translating human genotypic information into patient care is in fact now. And so we need to move on to addressing the downstream bottlenecks. How we've to assemble the infrastructure to do the work I'm going to describe today. We've assembled a facility at Pharmaceutical Sciences, at the Pharmaceutical Sciences Center, which includes 2 Illumina HiSeq instruments, the 2013 generation instrument and the newer 2014 instrument that uses the Illumina's version 4 chemistry. And we also have an additional Illumina MiSeq and extensive automation to process samples. And I'm not going to emphasize any more the infrastructure because the platform I'm going to discuss for analyzing the data is really agnostic to the devices used to generate the data. And if we looked at this slide 2 years from now, it would almost certainly showcase different instruments. So that's the infrastructure, and I just want to step back and remind everyone that the next challenge is managing the data, and it's important to remember that when we're talking about the human genome, we're talking about an awful lot of data. Each genome is about 3 billion bases, and if you want to do the math as represented on this slide, a single human genome can be represented by 1.5 gigabytes of space. That 1.5 gigabytes doesn't come out of whole cloth. Rather, the raw sequence data requires about 700 gigabytes of data, which is reduced following alignment to about 300 gigabytes. The variation data is a much smaller amount of information. And finally, we get down to a manageable file size, including which encompasses each individual's SNP profile, which is about 3 million polymorphisms per individual. So as you're well aware, between any one of us, there's about 3 million different genotypic variants. When you then go, you take a big hit when you go to ask how many of these 3 million variants are actually clinically testable. And that number is quite small at present. As next generation sequencing data becomes implemented in the clinic, the number of clinically testable variants will undoubtedly rise, but it's not clear that the rise in clinically testable variants will see a concomitant increase in the number of clinically actionable variants. Right now, you can, depending upon whom you ask, the consensus is that the number of clinically actionable variants, that is, a variant detected in an individual that will lead you to make a therapeutic decision, is really rather small. And it's something of a catch-22 in the sense of the number of clinically actionable variants depends on the quality of our information regarding the frequencies of alleles in a population, and consequently the deviation from those normal frequencies in any one individual. And so you can't improve upon those statistics unless you collect more information in the form of more human whole genomes and more human whole exomes. And in order to do that, we've decided to implement, just to step back a second, the individuals who we've, in our study, we've designed to deliver this information and interpret this information are practicing community pharmacists. The reason being is that these individuals are expert at interpreting and delivering information regarding a whole host of physiological functions, such as kidney function, how that relates to drug therapy, liver function, how that relates to drug therapy. And we're now equipping community pharmacists to be able to deliver information regarding gene function and how that relates to drug therapy and drug efficacy. This slide here is somewhat of a straw man. The gene function report is not going to look like this. And another way people have delivered genetic information looks a little bit like this 23andMe report. But again, this is not the kind of report that will be either readily understandable in a clinical situation, or in the case of our study in the community pharmacy. So we need something much more interpretable. And here's where the GNOME solution will come into play. And so I just want to walk you through how we're going to, this is the 30,000-foot view, how we're going to use the GNOME platform to eventually go from aligned genomic reads, aligned to the reference genome, and get to a report that can be delivered in a community pharmacy setting to a client that's easy to understand and easy to interpret. So let me just walk you through this at a high level. At the front end, we enter aligned reads into the Gnosis platform, And these first two steps are really the heavy lifting that's done by the GNOME device. And so rather than having a team of bioinformaticists, we have automated these steps as much as possible. After the variants are called, the variants are annotated, and that is where the value is added to the variant data. And this is where both internal databases and external databases are queried to provide information regarding those particular variants. Then a set of samples or a set of individuals is used to query either a reference set or a different population of samples. The variants are filtered according to those comparisons, classified, and then the results of that query are ultimately interpreted into a draft report. And so you can imagine, and this is how we've designed our study, the human exome data, which will be collected, which will be derived from saliva collected in community pharmacists in BC, that data will then be queried by the GNOME system for whatever particular disorder we're interested in. So in a sense, the cost of the queries is high at the front end for the first test, but every additional test is essentially just an electronic in silico query. So each gene panel comprises multiple queries. Each query employs a number of different filters, and these can be toggled on or off depending on the level of resolution required. And each query also focuses on a set of targets, a particular reference genome or gene list or gene region, and can further be customized with respect to the type of comparisons done, whether it's this set of individuals versus a reference and the like. And then the end of the process is a report that includes the variant data, classification of that individual's variants, and then importantly, different action layers that can be investigated in a hierarchical fashion. And so the data can be presented to the client in a layered manner such that the education aspect of the interaction is emphasized, and one will only go on to the next layer after the first set of information is transmitted and understood. So that addresses the second bottleneck, and it is absolutely a work in progress, but it's one of the attractive features of having the GNOME system in place at the front end. Once we've collected a sufficient number of exomes and genomes, is that we can iterate our queries as the database of genomes and exomes grows. And you can imagine that the resolution of each report will improve as the statistical power of the database improves just due to organic growth. So the third bottleneck that we'd like to address is populating the database. And this gets back to a point I made a moment ago, and that is the idea of having the analytical tools for analyzing the data in place as soon as you begin to populate the database. And one could argue that you need to reach a threshold of data before the data has statistical significance. And that is likely true, but the fact is, in order to know when you've reached that threshold, you want to be iteratively querying the database and analyzing the data as you go. And so our argument is that both aspects should be built in parallel. And just to remind you of the obvious is that if the data quality is bad up top here, regardless of how good your model for predicting variance and their influence on phenotype is, your results will be of low quality. Along similar lines, if you have perfect sequencing data but bad models, you can't really expect anything but poor results. And so in our idealized world, we have high quality 30 to 50x human exome data analyzed using the Gnosis platform, and ultimately the results will be actionable clinical information that we can use in the community pharmacy. It's not going to be as simple as that. The theme here is the information will be layered, and the more layers we add to your genotypic information, the richer and better, more predictive the test can be. So the genotypic information will be added, will be combined with clinical test data, as well as phenotypic data collected at the front line by community pharmacists and combined with the genomic information. Further down the road, as technology advances, we'll be able to capture transcriptome data as well as rapid changes in metabolome data for each patient. So this is really looking several years into the future towards what a personalized medicine approach in the pharmacy will look like. And so, so we've identified, I hope I've conveyed to you some of the challenges that we've identified and how we're going to iteratively push those challenges and use that information to move towards our goal, which is to empower community pharmacists in the province of British Columbia to understand how genotypic information impacts whether or not a particular individual will experience a particular adverse event. So I'll go over in the next 15 minutes or so the overarching goals of our projects, some of the mechanics of the project, compare and contrast our efforts with other existing efforts, particularly in North America, and then describe in overview the different parts of our pilot project, and then finish with an eye towards the future. So this is a slide that many of you may have seen, and it emphasizes our overarching goal, which is to identify the potential for toxicity, reduced response before treatment. And in addition to that is to identify those members of the population that will respond best to treatment. So you know, in any population, you have a mixture of individuals, and if you could wave a magic wand and segregate them into these four groups, you wouldn't be giving the medication to those that can't benefit and can only experience toxicity. You would clearly be giving them the therapeutic to the subpopulation that will benefit without any toxicity, and for the groups that will benefit but will experience toxicity, you will need to use the genotypic information in as high resolution a way as possible. And ultimately, this will instigate the paradigm shift that has been discussed for many years, and that is rather than take a diagnosis, test a drug, observe the effect, and then repeat until the desired therapeutic outcome is obtained, use genetic information to assess the predisposition of a particular patient, inform the diagnosis, and then select the right drug, and again use the predisposition, the genetic information we have, to and combine that with close monitoring so that we can improve this paradigm over time. I do want to mention that our effort in community pharmacies is one of several. The promise of personalized medicine, which has been discussed for literally decades, is now, we're now witnessing the implementation of those concepts. There was a study published in 2012 from a collaboration with Kerr Drug in North Carolina in the U.S. It involved a single pharmacy. It was an interventional study using an anticoagulant, and I'll describe that in a little bit more detail in the next slide. A large pharmacogenomic study called eMERGE that involves 20-plus sites. Some are observational, some are interventional. Some of those sites are in clinical pharmacies, some are in community pharmacies. This is an ongoing effort that will certainly be yielding very interesting data over the next several years. And then there's our effort, which is called PRIME and is launching this year. So I mentioned the Kerr Drug Pharmacy study entitled Implementation of a Pharmacogenomics Service in a Community Pharmacy. It was published, I apologize for the typo here, it was published in the Journal of American Pharmacy Association in 2013, not 2003. And the main outcome measures of this study were patient participation, prescriber acceptance, the time it took for the test, and the outcomes of patient claims. And the results are, it was a small study. There were 41 patients who met the criteria. A little less than half enrolled and completed the study. 9 patients had a wild type genotype for the variants tested. 9 had variants at those positions. The pharmacist then made recommendations to modify the medication based on, in those 9 individuals. In all cases, the modifications were accepted by prescribers. 17 patients filed reimbursement claims. And the not so good news is that 5 were not billed due to technical issues, 12 were billed, and none of them were paid. So the conclusion is that the service can indeed be an extension of medication therapy management services in a community pharmacy. There were no insurmountable hurdles encountered during the implementation. And the prescribers were receptive, but I'm not exactly sure what receptive means here because reimbursement did not happen. So I think that the conclusions are fairly clear that reimbursement is going to be a challenge. We're not going to be addressing that for the moment. For more details about the Cur drug studies, I would point you to clinicaltrials.gov, and you can see pretty much all of the details about the trial design, the researchers, and their results. On to the next project, which, as I mentioned, is a much larger, an order of magnitude larger, or 2 orders of magnitude larger, 9,000 clients involved in distributed across 20 different centers. And rather than focusing on 3 or 4 genetic variants associated with a anticoagulant, the eMERGE project is focusing on 84 so-called pharmacogenes. These pharmacogenes defined by the eMERGE consortium comprise a number of classical pharmacogenes, including the genes involved in drug metabolism, drug import and export, as well as other genes that have been associated with particular cancer types. So here they are. Here are the 84 pharmacogenes in no particular order. Some of these will look familiar when I describe the genes that we're going to be focusing on in our study. It's important to note that the eMERGE project is focusing specifically on these 84 genes, and that is to say the genotypic information will be restricted to these genes. It's a gene panel approach. The exomes, neither the exomes nor the whole genome will be sequenced as part of the effort. So that's the sacrifice of this study, is it'll be restricted to these genes. The advantage of focusing on this particular cohort of genes is that using next generation sequencing, the depth of coverage of each one of these 84 genes will be on the order of several hundred to 1,000x coverage. That now leaves us about 10 minutes to describe our case study, which is goes by the acronym of PRIME, which stands for Genomics Registry for Individualized Brainchild of Dr. Ron Reed at the University of British Columbia, and a colleague of mine. And our two-pronged approach will focus first on collaboration to collect well-phenotyped patients, and then the second and really key element is that we're utilizing the trust and accessibility of community pharmacists as frontline healthcare for patients, and ultimately to deliver the patient information, the genotypic information back to the patient. And so one of our main emphases here is education, both education of the pharmacist and education of the client as to what genetic information can and what it can't do. So how are we going to do this? Well, we're taking this crowdsourcing approach, which is to sort of cast a broad net across the province and build, in a sense, build a population of individuals. And by that, I mean collect sufficient, and by sufficient, we will only know once we get there, but collect sufficient number of exome sequences to populate a database such that we can derive allele frequencies with sufficient statistical confidence, such that we can assess variations from the norm and then ultimately correlate those variations with phenotypic response. Sequencing, as I tried to make the argument earlier in the talk, makes sense now. It's cost effective, and it's particularly cost effective if you think about this as being one test with one upfront cost and a lifetime of amortization. And I'm probably preaching to the choir here, but it's fairly clear that the genome information allows for truly personalized medicine. Again, the point that you acquire the information once and query it indefinitely, and this includes querying for the same indication because as I mentioned earlier, as the database gets more populated, the resolution of a particular query will improve. And the adage that we'd like to put forward is that you sequence today. I have here for questions of tomorrow, it should really read sequence today for questions of today and questions of tomorrow. So when I said we're collecting data via collaboration, I can't emphasize enough that the value of your genotype is only as good as the depth of your phenotype. And so we have focused on genotyping or sequencing well-phenotyped samples. And that got an enormous boost through our collaboration with the BC Generations Project led by Dr. John Spinelli at the BC Cancer Agency and the University of British Columbia. And through this collaboration, we have access to now 32,000, not 26,000, well-phenotyped samples. So these are normal individuals, ages 35 to 65, that have filled out an extensive hour-long questionnaire regarding medical history and life history. And we're going forward with sequencing the exomes of these 26,000 individuals. And that's described in the next bullet. We're using these sequences to seed our database that will be further added to by saliva samples that we acquire in the community pharmacies. And I said that your genotypic information is only as good as your phenotypic information. You can make a similar statement regarding the quality or the utility of your data is only as good as the number of links you could make to that data. And along those lines, we're working closely to develop linking protocols with Population Data BC. So that's how we're going to populate the database via collaboration. We're also going to populate the database by action, and that action is going to be done working closely with community pharmacists in the province, and they're going to be collecting genotypic data in the form of saliva and phenotypic data in the form of questions to clients. So we've standardized our consenting process, which is under final review at the moment, and it's going to be a electronic consenting process using short videos that will be available quite soon. For describing genomic information can and can't do, and using the videos to educate the trial participants. We've opted to use saliva collection for our DNA samples. The quality of the data we've been getting from saliva collection is at least as good as that from blood draws. And then all of these activities are governed by standard operating protocols. So this slide is just evidence that we have SOPs for everything from collecting the saliva to shipping the saliva and delivering back the information. We have preliminary ethical approval for the study. And so in the last couple of minutes, I'm going to focus on the opportunity that we're going to emphasize in our initial pilot project. And the therapeutic that we're going to be looking at is warfarin. And there are many reasons for focusing on warfarin. For the purposes of this talk, we have the reasons behind selecting warfarin is that it does represent the gold standard for pharmacogenomics. It's one of the few therapeutics for which the FDA in the US has label guidance that instructs clinicians to seek pharmacogenomic information in determining initial dose, and the polymorphisms associated with warfarin efficacy have been known for several years. So, as I said, we're using warfarin here as a benchmark. The reasons, just a couple of the reasons, are highlighted in this slide. Warfarin efficacy is crucial to health. Underdosing and overdosing are both potentially very adverse events, as I just mentioned, there is the potential for severe adverse reactions due to underdosing or overdosing. It has a narrow therapeutic index, but within that index, it's quite effective. And as a consequence of its narrow therapeutic index, it requires close monitoring to titrate the dose. And that monitoring right now is done biochemically through blood draws. A little bit of warfarin history. It was first discovered when farmers observed the effects of a particular fungal metabolite acting as an anticoagulant, causing distress and death to their herds of cattle. A modification of the fungal metabolite was made by the Wisconsin Alumni Research Foundation, which gave this Coumadin variant its name of warfarin, and that was 1948. Soon after it was developed, this soluble and bioavailable version was developed. It was given to President Eisenhower after a myocardial infarction, and that definitely caught the attention of the medical community. It was approved a few years later by the FDA, and interestingly, its mechanism of action was not defined until 1978. The influence of genetic variation on warfarin efficacy, as I said, is quite well known, and polymorphisms in these two proteins, metabolizing enzyme CYP2C9 and the vitamin K epoxide VCORC-C1, account for the lion's share of the genetic variation. There's still an unknown component to the variation. Whether that's environmental or genetic remains to be determined. So we're at our plan of attack for this pilot is to collect saliva in community pharmacies, and from that same sample, we're going to sequence the individual's exome, and we're going to benchmark that same sample using a PCR-based allele discrimination test, a more classic approach that will interrogate these 18 warfarin polymorphisms. And this two-pronged approach has the advantage of the PCR test will provide benchmark data for which we can compare the exome data. And ultimately, the hope is that subsequent tests will no longer have to rely on the PCR benchmark, and we can use the exome information directly. one of the nice things and one of the key features of using this SNP panel for PCR is, as I said, it validates the exome sequencing. But by using the GNOME platform to restrict our initial queries to these very same 18 polymorphisms, despite the fact that we have the entire exome in the database, we immunize ourselves and the pharmacists against incidental findings. We make sure that the infrastructure, the protocols, and the delivery information are all in place, so that we walk before we run. During the course of this initial pilot, we'll work through the kinks of populating the database, and then, and only then, can we move on to looking at other gene-drug interactions. And it's a well-known fact that the FDA has characterized over 150 drug-gene interactions. This, despite the fact that there's only a handful that have label guidance on them. Of these 161 FDA drug-gene interactions, there are 141 drugs that involve 48 genes. And so you can imagine once the warfarin test is completed, benchmarked, and analyzed up to interrogating these 161 drug-gene interactions using the same data that we've collected and the same GNOME interrogation platform. So just to recap, the first report will involve these 18 warfarin polymorphisms. Our second report is going to be a much more inclusive report interrogating the 161 FDA drug-gene interactions. And then the third, fourth, fifth, sixth reports, you can imagine we will be looking at every new compelling drug-gene interaction that comes online. And you can imagine in between here and here, the second report and the ninth report, we'll be benchmarking ourselves against the eMERGE pharmacogenes to be able to compare and contrast the data we collect with the eMERGE data. So I'd like to apologize for going a little bit long, but I do need to stop and at this point and thank our partners. The UBC Pharmaceutical Sciences has given unqualified support to this project in the form of space, equipment, and cheerleading. UBC Infrastructure Technologies has provided support in innumerable ways. The BC Generations Project, as I mentioned, John Spinelli's group and Trina McDonald have provided samples and advice. The BC College of Pharmacists and the BC Pharmacy Association have been great partners. The Canadian Foundation for Innovation has provided a lot of funding for the infrastructure, and we've been working with our vendors, Illumina, on the sequencing side of things to streamline our protocols, and Microsoft to lock down a simple delivery system for the genetic information in the pharmacies. And of course, to GNOME for helping us build these panels and learn the power of the platform. And so with that, I'd like to thank you and let you know that any and all feedback is most welcome. And I believe the folks from GNOME will provide contact information, but please don't hesitate to contact me at my email at the university if you're local to use and test out the technology. and if you just have questions, we welcome them. So with that, I'll stop and I guess take questions.
Thank you, Corey, for this excellent talk. Everyone, you are welcome to type questions into the question pane of the GoToWebinar control panel, as some of you have already done. As I mentioned earlier, this presentation has been recorded, so we will be sending you out an opportunity to get a copy of the slides and a link to the recording. I'd also like to announce, before we get to questions, that our next webinar will be held on August 14th. I'll be the presenter for that webinar, and I'll be speaking about further about the concept of in silico panels, specifically how panels like the ones Dr. Nislo has been describing are being implemented in Gnosis. And I'll demonstrate some of these specific examples on the Gnosis and show you how a new one can be rapidly put together. Jessica will send out an invitation to all today's attendees for that webinar. So on to the questions. A first question that came in was about novel variants or variants of unknown significance that you encounter when looking at these pharmacogenes, and how you'll be addressing those in this project that you're doing.
Oh, that's a great question, and actually one I didn't anticipate. Because I was emphasizing how we're so perfectly immunized against information we won't know how to interpret. But if we come across, and it's probably not unexpected, variants of unknown significance in the very genes that we're interrogating, we're going to consider them in a separate bin, and we're going to collect the information in terms of how frequent they are and how rare they are. But constrain our 18 particular variants we've defined in the pilot, and consider the variants of unknown significance in the genes we're interrogating, as I would we could call them slightly incidental findings. So they're clearly, or they may be actionable, but we're going to cordon them off for the purposes of the pilot.
Thank you. So this is a question about the eMERGE project, which realize isn't what you're doing here. But you know, when you talked about the 84 pharmacogenes that they're looking at, do you know whether they're covering all of FDA's recognized, validated biomarkers and gene-drug interactions?
You know, my inclination is to say they do, but I actually don't know that for certain. It's definitely a large subset of the 48 genes the FDA has identified, but it also, interestingly, includes some of the genes that have been defined as incidental findings that should be reported. So rather than giving misinformation, they have several excellent white papers out, not about the data yet, but about their approach. And so I would refer the questioner to the eMERGE website.
Yeah. Okay. Thank you. So let's talk about your warfarin project. Maybe this should be obvious, but the patients that you're looking at, are these individuals for whom warfarin has been prescribed, or is this more of a general population study?
It's an all-comer study. We will the pharmacists will be looking and emphasizing. There's limited bandwidth in every pilot, so pharmacists will be favoring patients who have been prescribed warfarin. But I do want to emphasize that as important as the warfarin data, equally important is the training and the implementation of the SOPs in the pharmacies and the return of that information. So you could call it a hybrid focused all-comer study, in that if it's not a requirement that you be prescribed warfarin, because this is a retrospective, not an interventional study.
So are these participants, will the data that you're generating on their exomes, including these warfarin models, would they be available in the future should they be prescribed warfarin or for any other reason?
Absolutely. So as part of our consent form, the individuals are consenting for future tests for future medications.
Terrific.
Including warfarin.
Yeah. So and how conveyed to the pharmacists so that they can use it? Is this directly in their systems? Is a static report or some other mechanism?
Great question. So initially it's going to be a static report because that's how the pilot was designed, but that's obviously not scalable and also not the best use of information technologies. So we're actively looking at the best ways to incorporate the results of these tests into in British Columbia. We have an excellent electronic medical record system for pharmacies called PharmaNet, and which includes a patient's entire medication history. And the way this information will be most valuable is if when the pharmacist goes to query PharmaNet, a flag comes up and says their genome is available for querying for a particular test. And so when I mentioned the data will only be as good as the links, this would be an example of the data will be orders of magnitude more valuable once it's in the patient's PharmaNet profile.
Well, and just for fun, let's wrap up with kind of a biological question. So what about epigenetic control in these pharmacogenes?
Aha. So in terms of right now, I'll take a stab, Ben, and you can maybe redirect. So are you asking about methylation variations in the genome? Because we're not capturing that right now. Or are you talking about epigenetic control of expression of these pharmacogenes?
I think any and all of the above. So basically stuff that doesn't come straight out of the genomic DNA sequence. And whether that's something that you're going to be addressing or how important it is, is that a big piece that's missing in terms of being able to do the most accurate prediction and prescription?
Well, absolutely, it's an essential part, and depending on who I talk to, control of gene expression is either considered a piece of genotypic information or a piece of environmental information, in that the cellular environment and the stress that a cell is put under can be considered an environmental change on gene expression. And so right now, by focusing on exomes, you do collect limited non-genic sequence, but I think, and I don't know that anyone would argue that the complete genome sequence will be much more informative. We won't be able to get direct expression data, but as the information and the inferences that are possible with respect to predicting gene expression from promoter and terminator and enhancer architecture is getting richer every day. So I think just from whole genome sequence, there will be a fair amount of predictions one can make on the levels of expression in a particular individual. That said, nothing will substitute for direct transcriptome data. And I think if we have this discussion 3 years from now, it'll be considered obvious that data needs to be included. I think we're going for the lower hanging fruit right now. And after we have the transcriptome conversation, the metabolome transfer conversation follows right after that. So, so this is a first step.
Okay, great. Well, thank you for answering all these questions. There are a couple we weren't able to get to today, but we'll respond. Thank you all for coming. Thank you, Corey, for this informative presentation. It's great to see the excellent translational work that's being done here. So again, thank you all for coming, and goodbye.
Thank you.